As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damages and infections, it also helps in cell-to-cell interactions and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactons, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectin, and protein. The cell wall of a young plant cell, the primary cell wall, is capable of a growth which gradually diminished as the cell matures, and secondary wall is formed on the inner towards membrane side of the cell. Para contain important point. Non-cell wall, non-living rigid structure form in plants and fungi. Algae have cell wall made up of cellulose, galactons, mannans, and minerals like calcium carbonate, while in other plant it consists of cellulose, hemicellulose, pectin, and protein. Cell wall of young plant cell, primary cell wall. After maturation, it gets diminished. Then secondary cell wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly is calcium pectate which holds or glues the different neighboring cell together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Para contain important points. Middle lamella is a layer mainly of calcium pectate. Cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of membrane organelles is distinct in the term of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum (ER), Golgi complex, lysosomes, and vacuoles. Since the function of the mitochondria, chloroplast, and peroxisomes are not coordinated with the above component, these are not considered as part of endomembrane system. Para contain important points. Endomembrane system include endoplasmic reticulum (ER), Golgi complex, lysosomes, and vacuoles. Endoplasmic reticulum (ER), electron microscopic structure of eukaryotic cell, reveal the presence of network or reticulum of tiny tubular structure scattered in the cytoplasm that is called the endoplasmic reticulum (ER), figure 8.5. Hence, ER divides the intracellular space between two distinct compartments. It means laminal inside ER and extralaminal means cytoplasm compartments. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum (RER). In the absence of ribosomes, they appear smooth and they are called smooth endoplasmic reticulum (SER). Para contain important points. Endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum (RER). Absence of ribosomes, they appear smooth, called smooth endoplasmic reticulum. RER is frequently observed in the cell actively involved in protein synthesis and secretion. They are extant and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid in animal cells. Lipid like steroidal hormones are synthesized in SER. Para contain important points. RER is frequently observed in the cell actively involved in protein synthesis and secretion, and extensive and continuous with the outer membrane of the nucleus. Smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal, lipid like steroidal hormones are synthesized in SER. Golgi apparatus. Golgi 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or crystals of 0.5 micrometer to 1 micrometer diameter. Figure 8.6. These are stacked parallel to each other, varied near to Cristernia are present in Golgi complex. The Golgi Cristernia are concentratedly arranged near the nucleus with a distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging material to the delivered either to the intracellular targets or secreted outside the cell. Materials to the packed in the form of vesicles form the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. The number of protein synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternia of Golgi apparatus before they are released from its transphase. Golgi apparatus is the important site for formation of glycoprotein and glycolipid. Para contain important points. Material to be packed in the form of vesicles form ER fused with a cis phase. Golgi apparatus is the important site for formation of glycoprotein and glycolipid. Lysosomes. These are membrane-bound vascular structure formed by process of packaging in the Golgi apparatus. The isolated lysosomal vacuoles have been found to be very rich in almost all type of hydrolytic enzymes, hydrolase like lipase, protease, carbohydrase, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acid. Para contain important points. Lysosomal vacuoles are very rich in almost all type of hydrolytic enzymes like hydrolase, lipase, protease, carbohydrase, optimally active at the acidic pH. Vacuoles. Vacuole is the membrane-bound space found in the cytoplasm. It consists water, saps, excretory products and other material not useful for the cell. These vacuoles is brought by a single membrane called tonoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other material against concentration gradient into vacuoles. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Para contain important points. The vacuole is the most membrane-bound space found in the cytoplasm. It consists of water, saps, excretory products and other material not useful for the cell. The vacuole is brought by single membrane called tonoplast. Amoeba contractile vacuole is important for osmoregulation and excretion. In Protista, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, single mitochondrion, useless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane-bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. It means the outer compartment and the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding called the cristia, single crista, towards the matrix. Figure 8.7. The cristia increase the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possesses single cellular DNA molecules, a few RNA molecules, ribosomes, 70S, and compartments required for synthesis of protein. The mitochondria divides by fission. Figure 8.7. Structure of mitochondria, longitudinal section, outer membrane, inner membrane, matrix, intermembrane space, cristia. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus impacting specific color to the plants. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for tracking light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like keratin, xanthophyll, and other are present. These give the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless pigments of varied shape and size, with stored nutrients. Amyloplast store carbohydrates, starch, example potato. Elioplast stored oil and fats, whereas the alveoplast store protein. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of Chlamydomonas, a green algae, to 20 to 40 per cell in mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of chloroplast is called stromata. A number of organized, flattened membranous sacs called the thylakoids are present in the stroma. Figure 8.8. Thylakoids are arranged in stacked like piles of coins called granum, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubes called the stroma lamellae connecting the thylakoids of the different grana. The membrane of thylakoid encloses a space called a lumen. The stroma of chloroplast contain enzyme required for the synthesis of carbohydrates and protein. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of chloroplast are smaller 70S than the cytoplasmic ribosomes 80S. Ribosomes are the granular structure first observed under the electron microscope as a dense particle by George Palladay, 1953. They are composed of a ribonucleic acid, RNA, and protein and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunit, larger and smaller subunit, figure 8.9. The two subunits of 80s ribosomes are 60s and 40s, while that of 70s ribosomes are 50s and 30s, whereas Swedberg's unit stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70s and 80s ribosomes are composed of two subunits. Figures 8.9 ribosomes. Smaller unit, larger unit. Cytoskeleton. An elaborate network of filamentous proteinaceous structure consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia single cilium and flagella single flagellum are hair-like outgrowth of the cell membrane. Cilia are small structure which work like arcs, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively larger and responsible for the cell movement. The prokaryotic bacteria also possesses flagella, with these are structurally different from that of eukaryotic flagellum. The electron microscope study of cilia or flagellum shows that they are covered with the plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. Axoneme usually has 9 duplets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as the 9+2 array. Figure 8.10. The central tubules are connected by bridge and is also enclosed by a central sheet which is connected to one of the tubules of each peripheral duplets by a radial stalk. Thus, there are 9 radial spokes. The peripheral duplets are also interconnected by linkers. Both the cilium and flagellum emerged from centriole-like structure called the basal bodies. Figure 8.10. Section of cilia, flagella showing different parts. A. Electron microscope. B. Diagrammatic representation of internal structure. Plasma membrane, peripheral microtubules, duplets, interduplets bridge, central microtubules, radial spokes, central sheet. Centrosomes and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both of centrioles in a centrosomes lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly shaped peripheral fibrils of tubulin protein. Each of the peripheral fibrin is a triplet. The adjacent triplets are also linked. The central part of proximal region of centriole is also proteinaceous and called the hub, which is connected with the tubules of peripheral triplets by radial spokes made up of protein. The centriole form the basal body of cilia or flagella and spindle fibers. They give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as easily as 1831. Later, the material of the nucleus stained by the basic dye was given the name chromatin by Fleming. The interface nucleus of a cell, when it is not dividing, has highly extended and elaborate nucleo- nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nuclei. Single nucleolus. Figure 8.11. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer, called the perinuclear space, form a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores of the pages, though which movement of RNA and protein molecules take place in both direction between the nucleus and the cytoplasm. Normally, there is only one nucleolus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect name of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example, erythrocytes of many mammals and sieve tubes cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm contain nucleolus and chromatin. The nuclei are spherical structure present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm. As it is not a membrane-bound structure, it is a site for active ribosomal RNA synthesis. Larger and more nucleolus nuclei are present in cell actively carrying out protein synthesis. Figure 8.11. Structure of nucleus. Nucleoplasm, nucleolus, nucleopore, nuclear membrane. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contain DNA and some basic protein called histones, some non-histone protein, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among these 46 means 23 pair chromosomes. You will study the detail of DNA packaging in the form of chromosomes in class 12. Every chromosome, visible only in dividing cells, essentially has a primary constriction or centromere, on the site of which a disc-shaped structure called kinetochore are present. Figure 8.12. Centromere holds the chromatids of chromosome based on the position of centromere. The chromosomes can be classified into 4 types. Figure 8.13. The metacentromic chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric centromere, the centromere is situated closer to its end, forming one extremely short and one very large arm, whereas telocentric chromosome has terminal centromere. Figure 8.13. Types of centrosome based on the position of centromere. First, metacentric, satellite, secondary constriction. Second, submetacentric, one short arm, one large arm. Third, acrocentric. Fourth, telocentric. Sometimes a few chromosomes have non-stained secondary constrictions at the constant level. These give the appearance of a small fragment called the satellites. Microbes. Many microbe bound minute vessels called microbodies